So thank you for joining us this afternoon. My name is Alex Chin. I'm a scientific partner with Medical Scientific Affairs with Roche Diagnostics US. Today I'm honored to be joined by Dr. Victoria Zhang from the University of Rochester, who's currently professor of the Department of Pathology and Laboratory Medicine, vice chair of Enterprise Strategy, and director of the Clinical Mass Spectrometry and Toxicology Laboratory, along with clinical chemistry at the University of Rochester Medical Center. So thank you for joining us, Dr. Zhang.
Right.
Yeah.
Thanks for having me. It's a great pleasure to be here.
Yeah, so today we're talking about something that's very passionate with laboratory professionals, right? So the role of mass spectrometry in patient outcomes. But particularly, we could maybe go over the first slide. Basically, what is mass spectrometry?
Yeah, well, that's a very good question to start the panel here. Mass spectrometry, when I think about that, I really think of this as a platform. So there's several parts. So I will start, as the slide shows, I will start for mass spec, since we call that mass spec, right? So what is the mass spec? So mass spec, for one, is short for mass spectrometry. So don't have to see the whole word, just see mass spec, it's fine. And there are different kind of mass spec. When we see mass spec, it's not just one thing. It's actually a group of various instruments. But they all commonly have three parts. One is the mass analyzer, which is to get the compound ionized. And then we have the mass analyzers, the ionizers, and then mass analyzer to analyze the mass to charge ratios, and the detector. So detectors typically, they're not much to talk about, but a lot of differences with how do we get the compound ionized, the ionizer, and analyzed. So here we're showing you here, as most people see, as a triple quad. So when we think about the mass analyzers, right, triple quad is one thing that we hear a lot. Actually, time of flight is another one with analyzer. The goal is the same, to analyze the mass over charge ratio. All right, so before that, mass spec has one unique feature. That is, it can only analyze compound that is being ionized. That's why the title also see the impact with the ions. So to get them ionized, we need a mass analyzer. We have the ionizers. So electrospray ionization, that's what people normally see. MODI is another one. Those are the soft ionization methods. There are several others. I think MODI and ESI are the most commonly used. So those are mass spectrometry portion of that. But for mass spec to work and function, we also need a part before that. That is chromatography. That's when people see mass spec like LC, mass spec, that could be liquid chromatography, or we can use the GC, that is gas chromatography, right, for GC mass spec. So the chromatography, their function is to separate the compounds. Let's just think about the body fluids, right? We have a lot of things in blood. So we're gonna have a lot of them to, for mass spec to be most effective, we have to separate them. So you don't want to, just shoot everything inside. So chromatography can help with that. But even before that, what we call that the sample preparation, right? I frequently get phone calls from the clinicians saying, hey, I have this compound in serum, can you measure that for me? Some of them, I'm like, well, it's not that straightforward. They're like, well, you can just shoot in your mass spec and do it.
Right.
I'm like, well, before that, we normally do, preparation. We have to figure out the chromatography, all the parameters. So most people just think, hey, I got something I can just inject. Well, it's not that easy. So that's a part of kind of the, what we do at the profession, right? That's why we kind of the platform for the mass spec to be able to work in the lab, right? It does require a lot of expertise and functions that for it to kind of fulfill its needs and requirements. So that's why we have here to show sample preparation, and then go to the chromatography, and then mass spec portion of that. And afterwards, there's still a lot of work need to be done. That's, for instance, peak integration. How do we know the system is working? QC, right? Calibration if we wanted to do quantifications. So anyway, so I can even speak a lot more, but at least overall, this is the overall workflow that we have, use mass spec as a platform.
Okay, thank you for that elaborate description. So, yeah, essentially, like we see on the slide, like 4 different
Right.
discrete steps, but
Different
you could
parts.
probably break them down even further. But
Yes.
you really alluded to really the challenges associated with a manual type of process, as much as we could probably get even semi-automated if in many today's modern labs. But those
Yeah.
are the really challenges that you face. What part of this do you think will be the most challenging for mass spectrum?
Yeah, that's a good question. As multi-stepped, manual and labor-intensive process,
Right. Yeah.
I would see, what began with the end in mind, which is for mass spec to be happy. We have to really start from a sample preparation. I always see that sample preparation is the most challenging part. If your sample preparation is done well, the rest becomes easy. But at least in me, when I work with my lab and talk to people, I feel like the sample preparation need to be well done. And frankly, that part is also the one takes the most the manual labor process.
Right.
Yeah.
Okay. Thank you for that.
Sure.
So we talked about mass spec, and another powerful method we have in the clinical laboratory is automated immunoassays. And so, obviously, these two potentially there are both powerful tools that we have in the toolkit for laboratorians. But maybe go over what are the advantages or disadvantages of mass spec and automated immunoassays.
Yeah. Great question. Those two compared constantly, right? Do I use mass spec? Do I use immunoassay? And why do I use them? I will see, just like the Swiss Army, right? The knives. You wanted to have a different kind of tools in your toolbox. I think that's the same way I think about mass spec and immunoassays. They are really complementary to each other, right? So for immunoassay, a lot of them in the core labs, we already know, even have the analyzers just behind us. That's very easy to use. You can have random access and very high throughput. You put the samples there, you walk away. And there's a lot of support also from the vendors. I know you guys done a phenomenal job when you help with the customers, even getting FDA approved assays. Majority of them, I think, are FDA approved assays on the analyzers. So I think that provide a lot of confidence and ease of use for the customers. It's like over the shelf, you can do it. So, but on the other hand, for the immunoassay, there are some things that cannot be done or cannot be easily done, or it doesn't have enough volume or need for a vendor like you guys to work on it. Right? You have to have ROI for your own, at the assays. So there is a gap between the patient's needs versus what the immunoassay could provide. Right? On the other hand, some of the immunoassay we know is antibody-based. With the antibody, even though it has monoclonal antibodies, there's still some interference that's involved with the assay. So there's another challenges that we have with the immunoassay. So I'll see as I talk about the limitations with the immunoassay, that's on the flip side, I think provided the opportunities for mass spectrometry. All right, so number one, with the mass spec, with the specificity. It's very specific. Immunoassay might be kind of a group of compounds. Drug abuse is a good example, right? We also the class of a compound, right? Opiates, you don't see, I get the identify morphine or different compounds. It's the class. So in a way, it's very efficient. But on the other hand, you miss the opportunity to identify individual compounds. So mass spec provided that. And also to some levels of sensitivity, which is you can measure very low level of compounds much better with mass spectrometry. So those are another very good examples of that. Another one I think I wanted to see is the multiplexing. With the mass spec, we know the MRMs, which is multiple reaction monitoring, which is a very important feature for the quadruples. So we cannot just measure 1, 2. Many assays have 10, at least for my own lab, like 20. I mean, we can do, technically speaking, if you allow your chromatography long enough, you could go like hundreds of compounds within one significant run. So that's a very powerful technique that for us to use.
Yeah. So very good, thank you. So essentially, if you take these two methods and employ them together into a clinical laboratory, you do have a really almost a perfect marriage, if I tell you.
I hear you.
Right,
yes, I agree.
For patient care.
Yep.
So essentially, we're talking about the specificity, right, of mass spectrometry, and can you tell us a little bit of your experience at the University of Rochester and how it has been employed for patient care and patient outcomes?
Yes,
Yeah.
yes, absolutely. We have a very big core lab. Being upstate New York, we're essentially I think I would like to think we're the largest academic medical center across at Western New York. We also have 7 affiliated hospitals. Part of my function as the vice chair is to integrate all the clinical services together. So we see patients like hospital from 12 beds to like over 900 beds of hospital, kind of anywhere in between. So mass spec actually provided a great value for our system. Being in the system, that means people, hospitals send samples to us. This is a picture that we have on I think on this side. I did not take a picture before we moved in. So you can still see this is a real life. We have all the things on the bench. We have the samples there. But this is actually our new facility. The hospital realize the value that we have to the healthcare, which had to have a brand new off-site facility for us. So those provide a lot of great value to the system. I can just give some examples. For instance, like immunosuppressant drugs, right? That's one of the common drugs that people use. We actually, see that's probably one of I call that fast turnaround time assays. We have same-day turnaround time mass spec that's provided, we do hundreds of testing every day. The other one, vitamin D, I think everybody already know that. We do that as well. Drug of abuse, we have that too as a confirmation, and we also have a large panel for screening, some endocrinology testing as well. We do have a whole strain, we have a reasonable size of test menu on mass spec. So all of them, I think, provide a great value for our patients. So another one I do wanted to mention, as you see the picture here, when I designed the lab, this also got the opportunity. You don't get many chances to design your own lab in your career, but this is one of the opportunities I had. So when you think about mass spec, it's not just an instrument. You cannot just wheel that in and it's gonna work. Right? It takes a lot of infrastructure. For instance, it needs nitrogen, right? And it takes a lot of power. When we think about the power, 110. So literally during the design, that's one of the architects talked to us, hey, we wanted to review, how many mass spec do you have? I'm like, okay, I have a dozen. Okay, I have a dozen plugs for you. I'm like, okay, so tell me where they are. I'm like, oh, here. I said, okay, this sounds really good. What's the voltage of that? They're like 110. I'm like, okay, let's need to change that to 220. And then on the other hand, I'm like, well, I think there are 2 or 3 plugs for each mass spec. But it did not stop there. Why don't you do the HPLC too? I'm like, oh, we can count that, with 1 plug, 1 power supply. I'm like, why don't you take a tour to the lab? I counted, it turned out so there's 12 or 15 plugs for 1 HPLC. The person literally stood there like, I think I need to go back and talk about the budget. They did not plan on that. This is just an example to see there's a lot of things we need to think about in mass spec. But also one thing I asked them is, you can see here, we don't have nitrogen tanks. I'm like, okay, I don't care where you put it. I want the nitrogen tank outside of the lab. As of today, I refuse to ask where the nitrogen tank is. So a lot of things there's many things need to be considered for mass spec. So on the other hand, I think all of this are worth it. Given the stage we have with the technology and the value that it provides for the healthcare, we tolerate this in a way, right? I would love to have something better, but this is what we have. We have to work around it. But all of this, I wanted to give people the right image of mass spec. It's not like it can do a lot of things, but on the other hand, it takes a lot of expertise.
And I find what's striking about this image here is that you have a whole lab of mass spectrometers,
Yes.
right? And so the space requirement, the infrastructure, and second, you're also talking about turnaround time, right? So operational efficiency. So the reason why you have so many mass spectrometers is because you have a downtime or what happens, right? So can you speak to a little bit of that and what?
Yes. Downtime is challenging. I would love to have backup for all the assays. we cannot afford it.
Yeah.
I can tell you we cannot afford it. We do strategically back up like assays, and some of them we have to back up probably on different instruments, split them up. We have to be creative. But on the other hand, like for Roche, we have Roche too. Everything redundant is not a problem because its availability is there and the function is there, and it's just you can relatively easier and cost effectively purchase them to fulfill that need. Right? But for us, we don't. We need to. We do have some. We strategically place them. Also, to have full backup, we're not there yet. So.
Yeah. Right. So, kind of leads into the role of mass spectrometry in clinical practice guidelines. So we have some images here, or just screenshots of various guidelines. It's probably not exhaustive, but you can see there's various areas or disease areas where mass spectrometry is employed. What are your thoughts on what can we do to get, or can we do more for patient care and work with clinicians regarding mass spectrometry?
Yeah, that's an outstanding question. Obviously, we spend money for a purpose, right? It's only how can we justify the purpose. The patient care is the purpose. I always say, $500,000 instrument is not the issue. It's only the issue if I cannot see how the benefit, first of all, money-wise, and the patient service perspective, right? Even something, hey, I spent $10 million, I can make $20 million. Well, that's pretty cheap, right? But it's not the absolute amount. It's how much value we can bring to the system. I think mass spec is that expensive toy, but it can bring a lot of value. I think those give great examples from direct patient impact. But at the end of the day, right, can we provide sufficient services and cost-effective options for the patients so that they can get what they need under a reasonable cost, right? So when I see this, I kudos to the group that have done a lot of things, guiding the group to choose mass spec as a choice. I have to say I would say there's a lot more possible applications of mass spec than what we see now. People probably even use endocrinology as an example, right? Should you use testosterone or even free testosterone or testosterone for women? Should that be on mass spec? Should that be on immunoassay? Well, you can do that on immunoassay, but it's just not accurate, right? So those are things the providers have to be educated. We constantly do this. I go around with the endocrinology group, and we constantly have, every year they have residents, they invite me and specifically talk to their residents about mass spec so that they can order the right test. But that's not true everywhere. Right? But I think there's still a lot of value, and those paper and guidelines really give people the opportunity to understand the value of mass spec. But as I see those lists, for instance, microbiology has been used, mass spec, right? I think there's probably some guidelines or to be there, evolving group as well. Proteins targeted mass spec start to show. More instead of small molecules, we start to do proteins and large molecules. I think there's room for us to further, work on expand the functions of mass spec too.
Okay, and so basically it seems like a collaboration of clinicians and in the laboratory for really, even though it costs a lot, but really the patient value and the benefits downstream is kind of really driving that, the collaboration, also bringing in mass spectrometer, even though it's an expensive piece of technology.
Absolutely.
Okay.
Absolutely, yeah.
Right, thank you. So, in closing, what makes you excited about mass spectrometry? What does it do? We see on this slide here that there's different, cohorts where immunoassays and mass spec can bring to the table. What makes you excited about mass spectrometry?
So many things. When I think about mass spec, I'm excited. There are a lot of things we can think about for mass spec. Number one, right? As I mentioned, I don't think mass spec is going to take over the world. As people know, I started the mass spec and separated science division. When at the time people say, hey, our goal is to go on the mass spec to take over the world. I'm like, that's a great idea, but I don't think that's good for patients. Right? If anything comes cheaper, why would we use something more expensive? Right? So the idea is that we use the right tool for the right purposes. So when I think about this, I really think the mass spectrometry and immunoassay are complementary. So we just have different tools for the right purpose and the right assay that we're going to use. You're starting with a call lab. I like the graphic that you put together. When we think about the patients, that's the center, right? What does the patient need? And what kind of assay and on what platform can fulfill the patient needs? I think that's the fundamental question we have to ask, right? So a lot of the immunoassay come to the first answer. But on the other hand, I think mass spectrometry going to come to play. For one, is that even with the current platform, we need to think more, like, are we using the right platform? I really think mass spec can be utilized more as a whole for our society. And secondly is, how do we mass spec even more accessible to the community? That's something I've been working on for the last few years. One is educating people. What can we do with the mass spec, right? Another is so that for people to be easily set that up and for what they do. Some they don't even have that in-house or become too expensive to send out. Something become cost prohibitive. I mean, we all learned what that means when your bottom line is red, right, during COVID. That's pretty scary. So that becomes a challenge. But on the other hand, I think if we could make mass spectrometry more streamlined, more automated, I'm going to put the word there, more automated, because I really wanted to see the automation of mass spec. I've been reading and talking many things on that. I think that's going to be a game changer. Get people accessible, and just sitting right next to immunoassay becomes a choice for the patient needs rather than becomes a choice of can we afford it. So.
Yeah, that's a perfect, I think, end to this this presentation. Really, the accessibility, right? And I think that really resonate with labs that have little mass spec or maybe no mass spec experience, but even to those that have mass spec experience, and also, I guess, expand it to other areas of the clinical laboratory.
Yeah.
Okay.
I'm very excited about that.
So thank you. I think we'll just end this the session, but any questions from the audience?
If you have a question for Alex and Victoria, we'll bring a microphone around to you. Feel free to just raise your hand. Thank you.
Well, if not, can I just add one more thing?
Sure, yeah.
All right.
Yeah.
I think another thought I have is, when we think about the future, right? So the current workflow, I use drug abuse as an example because many people do it. That's kind of one of the largest assays that we have. The current workflow is we use immunoassay as a screening. And then for the positives, we use mass spec as a confirmation. There's many considerations to this. So just think about it. If mass spec is as easily accessible as immunoassay, as cost effective as immunoassay, why do we need the first step? Right? Wouldn't that be nice?
Yeah.
Right? So we can take that out of a lot of, save some time for the patients, save some money for the patients. You go directly there to the right testing. So I think that's, great. And another is I use testosterone again as an example. What happens is many times we run immunoassay and the provider realizes, oops, we got the wrong assay. Most time is they don't have that easily accessible, right? The same way is here, if we have mass spec easily accessible and just right there as cost effective as immunoassay, and basically the cost is going to be out of the picture. And people just go, hey, we have immunoassay, we have women, right? And we should automatically reflect that to mass spec because that is the right assay for this test. So rather than we think about, okay, how much does that cost, right? So that's why I'm also thinking about putting mass spec right next to the immunoassay analyzer is going to become so many potentials we're going to have for our own laboratory and also for patients.
Yeah, good point. I think regarding the quality management process and how much it costs for an automated immunoassay, but the the security of mind to have that whole, start to finish being all kind of taken care of instead of going piecemeal, let's say for a manual process to take care of that quality management process. It could be quite daunting and a lot labor intensive, right?
Yes,
So, yeah.
absolutely.
Yeah.
Yes.
Any other questions? Okay, well, I think we'll close today's session.
Yes.
Yeah.
Thank you, Alex and Victoria. Let's give them one more
All right.
round of applause.
Okay.
Thank you very much for this meaningful conversation.